Hello, my dear students. Hope the exam was good. Let's discuss the paper Zoology Section A from 151 to 185. Question number 151. Which of the following characteristics developed for the first time in the phylum to which Nereis belong? And you know that Nereis belongs to phylum Annelida. It belongs to phylum Annelida. Annelida are known for having a true metamerism, first time a true coelom, better developed circulatory system, nervous system, and all those things we know about Adler. They are metamerically segmented worms with true coelom. So, which of the following characteristics develop for the first time in the phylum? First time bilateral symmetry, if you see. Bilateral symmetry you can see for the first time in phylum Platyhelminthes, which are otherwise called ribbon worms, dorso-ventrally flat worms. So they are bilaterally symmetrical. First time bilateral symmetry you can see generally in case of Platyhelminthes, generally. So that is not the first time case. True coelom, yes. First time you can see a true coelom in phylum Annelida. Metamerism, segmentation, metamerism, a true metamerism can be first time seen in phylum Annelida. Therefore, for question number 151, answer is 4, that both 2 and 3. And you know that coelom is the body cavity which is lined by mesoderm. And coelom has its own functions. So for 151, answer is 4. Okay, with that, let us come to question number 152. 152, what is not true for a mollusks? So, segmented body, no, they have unsegmented body, having head, visceral hump, muscular foot, so this is not true. A rasping organ called radula, yes. With chitin, yes. Mantle cavity with feather-like gills, you know, ctenidium actually. So, feather-like gills which are ctenidium. So, gills with excretory function, yes. So, for 152, answer is 1. 1. With that, I come to page 153. 153, consider the following characters. Operculum, that is gill covering. You find operculum or gill covering in bony fishes or osteichthyes. You don't find them in chondrichthyes. Air bladder, yes. Air bladder, which is otherwise called swim bladder. Air bladder, which is otherwise called swim bladder, you can find in what is so called as bony fishes. Placoid scales, you will find, these are minute scales which are found in chondrichthyes. Okay, so this you find in osteichthyes, this you find in osteichthyes, this you find in chondrichthyes. So the characters that are present in bony fishes, you know, osteichthyes, which are otherwise called bony fishes. So A is present, B is present, C is not present. A, B, C, no, this is not proper. A and C, no. A and B only, yes, that is the answer. B and C only, no. So you know the chondrichthyes and osteichthyes. Chondrichthyes generally they are marine. Osteichthyes can be freshwater or marine. There are so many differences. You can find ganoid, cycloid, ctenoid scale in case of bony fishes and placoid scales in case of cartilaginous fishes. Heterocircle caudal fin you can find in case of chondrichthyes. Homocircle or difficircle caudal fin you can find in case of bony fishes. And reward differences in there. So for question number 153, answer is 3. You find operculum, generally you find in bony fishes. Air bladder or swim bladder to provide buoyancy you find in bony fishes. Placoid scales, these are minute scales of chondrichthyes So A and B only. So for question number 153, 3 is the answer. With that I come to question number 154. Homeothermy. Homeothermy, this is otherwise referred to as warm-blooded nature, warm-blooded organisms, is exhibited by amniotes. No, the question is, what are amniotes? Amniotes are those organisms which has extra embryonic membrane such as amnion, chorion, allantois. Amnion, chorion and allantois. And classical example include reptiles, birds and mammals. Reptiles, birds and mammals come under amniotes. So, you know reptiles, if you look at reptiles, these are poikilothermic or cold-blooded organisms. Cold-blooded or poikilothermic. All amniotes parameter. Homeothermy is generally exhibited only by birds and mammals. Exhibited by birds and mammals. Exhibited by aves and mammals. So first cannot be the answer because amniotes nu parayumbo reptiles adil padum. Reptiles cold blooded aanu, warm blooded alla. Poikilothermic aanu, homeothermic alla. Aves and mammals, that is the answer. All vertebrates, no, you know. Fishes, cyclostomes, amphibians, reptiles, they are cold-blooded organisms. The only vertebrate, birds and mammals, are warm-blooded organisms. Upper reptiles and aves, not possible. Aves, warm-blooded, but reptiles, cold-blooded. Therefore, for question number 154, answer is 2. Rand answer. Randan. With that, I come to question number 155. Large amoeboid cells that are part of our innate immune system. Innate immune system, you know, is non-specific defense, innate immunity or natural immunity or congenital immunity, non-specific immunity. So when you look at that, large amoeboid cells which are part of our innate immune system, cellular immunity also found in aerial tissue is definitely this is macrophage. Mast cells, you know, all these questions are directly from your NCERT. Read and learn your NCERT thoroughly, word by word, line by line, paragraph by paragraph, diagram by diagram, year by year. So this is a direct question. So for 155, 2 is the answer. With that I come to question number 156. Which one of the following is a protein deficiency disease? So if you look at muscular tetany, that is a parathormone problem. Osteoporosis is also associated with calcium metabolism and parathormone, calcitonin, homeostasis disturbance and all those things. So it's a hormonal disorder, that's what I want to tell you. So in muscular tetany, there will be muscular spasm. In osteoporosis, the bone will become weak. Kwashiorkor is a protein deficiency, malnutrition disease. So indigestion is because of no sufficient enzyme or all those things. So the answer for 156 is 3. Which one of the following is a protein deficiency disease? Kwashiorkor. And if it is protein energy malnutrition, marasmus. So 156, 3 is the answer. With that, I come to 157. Volume of air that will remain in the lungs after a normal expiration. That is what is FRC, functional residual capacity. That is ERV plus RV, you know. So the best option out of 4 is it's 2300 ml, which is a close number. Close number. So for question number 157, answer is 3. Okay, tidal volume, residual volume, oh, that's okay. So here it is FRC value, the closest value is 2300 ml. With that, I come to question number 158. Question number 158, the partial pressure of oxygen is equal in. Let us see. So partial pressure of oxygen in atmospheric is approximately 159 mm per Hg. In alveoli, it will be 104 mm per Hg. In oxygenated blood, it is supposed to be 95 mm per Hg. In deoxygenated blood, it is 40 mm per Hg. In alveoli, it is 104. Deoxygenated blood, it is 40, and in tissues, it is 40. So the partial pressure of oxygen is equal in deoxygenated blood and tissue. Answer is 4. All the values are in mm per Hg. So 158, answer is 4. With that, I come to 159. Partial pressure, slight 1 or 2, that you know. So the values now 159. 159, when only type B antigen is not present. So if this is your RBC, when only type B antigen is not there, so generally it is A antigen. When only type B antigen is not present on the surface of RBC of a person, his plasma would contain. Only type B ഇല്ലാന്നേ ഉള്ളൂ. അപ്പോൾ A ഉണ്ടാവാന്നാണ് പറയുന്നത്. അപ്പോൾ no antibodies possible അല്ല, കാരണം no antibodies you find in which blood group? You find in AB blood group. And for AB blood group, B അവിടെ ഉണ്ടാവണം. When only type B antigen is not present. അപ്പോൾ B മാത്രമില്ല, ബാക്കി ഉണ്ടാവുക. ബാക്കിയുള്ള ആൻറ്റിജൻ ഇതാ A. So next, only antibody A means B blood group. So that is not possible because here type B is not there. Only anti-B, correct. So this is A blood group. So that is what the question is also asking. So for question number 3, question number 93, both anti and anti-O, no antigen is present. No antigen is present. So here it is told only type B antigen is not present. Means type A can be present. So antigens, you know, A or B or AB in case of AB blood group. So here the best answer is 3. When only type B antigen is not present, that means type A is present. So this question is about A blood group. And the plasma would contain only anti-B. So for 159, 3, you know, universal donor is O negative and universal acceptor is AB negative. And you know these are sugar molecules which decides the blood group as A, B, AB, O. For A blood group, antigen A, antibody B. For B blood group, antigen B in plasma, antibody A. AB blood group contain both the antigens but no plasma antibodies. And O blood group contain both the antibodies but no antigen. So here for type A antigen, antibody B. So 159, 3 is the answer. And you know for both antibody A and B, this is O blood group. O blood group contain no antigen, but here question only B is absent, that means A is present. O blood group. Okay, so answer is 3. With that, I come to question number 160. 160. The cells that release heparin and histamine, body fluids and circulation, It is very clearly mentioned also. Basophil. They release the anticoagulant which is called heparin and histamine. Eosinophils, mast cells, neutrophils, neutrophils you already know, these are most abundant of all leukocytes. They are also called as polymorphonuclear leukocyte, that is another name. So for question number 160, in blood, in blood, it is basophil. So for 160, the answer is 1. With that, let us go to question number 161. What is used as an osmolyte? Osmolyte is salt water balance, homeostasis. ശരീരത്തിലെ ഉപ്പും വെള്ളത്തിൻ്റെയും ഒരു ബാലൻസ് നിലനിർത്തുക എന്നുള്ളതാണ് ഇതിൻ്റെ അർത്ഥം. What is used as an osmolyte in humans to increase the interstitial medullary osmolarity during the process of concentration of urine? You know from your NCERT, if you consider this as nephron structure, it's like this. So I can make this little bit thicker. So this is thick segment of ascending loop of Henle. This is collecting that. Small amount of urea is released into medullary interstitium, which is then taken by the thin segment of ascending loop of Henle, so that there is an osmolarity or osmolyte that is maintained. NCERT വളരെ ക്ലിയർലി ഇത് മെൻഷൻ ചെയ്തിട്ടുണ്ട്, ഡയഗ്രാം ലെവലാണെങ്കിലും. അപ്പോൾ ഇവിടെ യൂറിക് ആസിഡ് എന്നുള്ളൊരു കോൺസെപ്റ്റ് വരാൻ വഴിയില്ല, എമോണിയം ഇല്ല. So what is used as an osmolyte in humans to increase the interstitial medullary osmolarity during the process of concentration of urine? So here also the direct answer is urea. Urea from the collecting duct gets into medullary interstitium and then to thin segment of ascending loop of Henle, and there is a osmolarity balance. So question number 161, direct question, answer is 2, which is urea. With that, I come to question number 162. Atrial natriuretic factor, that is what ANF stands for. Atrial natriuretic factor, atrial natriuretic factor, or ANF it is written. Sometimes it is also written as atrial natriuretic peptide. So in some book you can also see this as ANP. So atrial natriuretic peptide or ANP, atrial natriuretic factor, ANF. So what is the role? So ANF promotes dash in the excretion of sodium. Higher the sodium, higher generally the blood pressure. So atrial natriuretic factor is antagonistic to renin, angiotensin, aldosterone system. അപ്പോൾ ANF അല്ലെങ്കിൽ ANP, it works opposite to renin, angiotensin, aldosterone system, which is otherwise referred to as RAS. അപ്പോൾ I can write very clearly here that ANF works antagonistic to RAS. You know that renin will act on angiotensinogen to convert angiotensinogen to angiotensin 1, and then angiotensin converting enzyme that will then form angiotensin 2. They all will increase the blood pressure. Aldosterone also contribute for the same. അപ്പോൾ ഇത് ANF, it decreases blood pressure. So ANF promotes increase in the excretion of sodium, thus dash and dash. So there will be decrease in blood volume and blood pressure. So here 1 and 2 are respectively, so increase and decrease. So for question number 162, 4 is the answer. There will be increase. And then there will be decrease. There will be increase and there will be decrease. Okay. So next question. Question number 163. That is also directly from NCERT. Which of the following has ATPase activity? Very simple. Head of myosin monomer. It's a direct question from your NCERT. That which of the following has ATPase activity? Answer is head of myosin monomer. 2 is the answer. With that, it's a direct question. Now I go to 164. So if you look at question number 164, human ribs are termed bicephalic. Question says the human ribs are termed bicephalic because. So bicephalic, 2 articulation, 2 ഭാഗത്തുനിന്ന് articulation surface ഉണ്ട് എന്നാണ് on their dorsal end. ഇതാണ് അതിൻ്റെ അർത്ഥം. So they have 2 articulation surfaces each on their ventral and dorsal end. അല്ല. They have 2 articulation surfaces on their ventral end. അല്ല. They have 2 articulation surfaces on their dorsal end. Correct. ഇതാണ് ആൻസർ. They have single articulation. അത് പറയില്ല, കാരണം ബൈസെഫാലിക് എന്ന് എടുത്ത് പറഞ്ഞിട്ടുണ്ട്. Bicephalic എടുത്തു പറഞ്ഞ സ്ഥിതിക്ക് you should understand very clearly that they have 2 articulation surfaces on their dorsal end. So for question number 164, 3 is the answer. With that, I come to 165. The brain stem does not include the. Appo you know that brain stem include your midbrain, pons orally, medulla oblongata, കൊടുത്ത ഓപ്ഷനിൽ. പക്ഷെ സെറിബെല്ലം ബ്രെയിൻ സ്റ്റെമ്മിൻ്റെ ഭാഗം അല്ല. അപ്പോൾ ഇവിടെയും ഇത് ഡയറക്റ്റ് ക്വസ്റ്റ്യൻ ഏകദേശമാണ്. So the brain stem does not include midbrain. Midbrain definitely it include. Pons orally it include. Medulla oblongata is also the part of brain stem. Cerebellum is not the part of brain stem. Brain stem include a part of diencephalon, midbrain, pons and medulla. So the brain stem does not include cerebellum. So for 165, 4 is the answer. With that I go to 166. 166 says oxytocin helping. You know, oxytocin majorly according to your syllabus, this is otherwise called birth hormone. Oxytocin is otherwise called birth hormone. So it helps in labor. So it helps in parturition or childbirth. This is one function that oxytocin help in childbirth. രണ്ടാമത്തെ ഫംഗ്ഷൻ ejection of the milk. Ejection of milk from mammary glands. Ejection of the milk from mammary glands. Post parturition. ഇതൊരു നോണോപെപ്റ്റൈഡ് ആണ്, അതായത് 9 അമിനോ ആസിഡ് അടങ്ങുന്ന ഹോർമോണാണ്. Oxytocin and vasopressin, they are synthesized in hypothalamus. And these 2 hormones which are synthesized in hypothalamus are stored and released via supraoptic nuclei and paraventricular nuclei from posterior pituitary, which is neurohypophysis. That means neurohypophysis only stores and releases the hormone. It does not synthesize. The hormones of neurohypophysis are synthesized in hypothalamus, but they are released from posterior pituitary. And oxytocin, you know, it is birth hormone. There are so many other functions also of oxytocin. So oxytocin is birth hormone which helps in child delivery, childbirth actually. And ejection of milk from the mammary gland. Production of the milk is under the control of prolactin. Milk production is strictly under prolactin. And of course, few of the other hormones, generally speaking, നമ്മൾ NCERT ആണ് follow ചെയ്യുന്നത്. Ejection of the milk from mammary gland is because of oxytocin. Now, with respect to that, implantation, no. Lactation and childbirth, that is the answer. Lactation but not childbirth, no. രണ്ടിലും അതിൻ്റെ ഫങ്ഷൻ ഉണ്ട്. ലാക്ടേഷൻ ഹെൽപ്പ് ചെയ്യും, മിൽക്ക് ഇജക്ഷൻ അതിനത് ഹെൽപ്പ് ചെയ്യും. ഫീഡിങ് ബിഹേവിയർ സമയത്ത് when the mother is nourishing the child, the child get the milk by the ejection from the mammary gland, and that is because of oxytocin. Lactation but not childbirth, no, it is not like that. Childbirth but no lactation, no, it is not like that. So for question number 166, 2 is the answer, lactation and childbirth. With that, let me come to question number 167. Question number 167, which of the following regulates carbohydrate, protein and fat metabolism in a manner opposite to that of insulin? So insulin, as you know, insulin is 51 amino acid hormone. 51 amino acid hormone. Insulin promotes glycogenesis, cellular uptake of glucose, and all these things. They are so which is antagonistic, which is opposite to insulin എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ glucagon, we know glucagon, alpha cells of the islet of Langerhans. Alpha cells of islet of Langerhans, they synthesize and release 29 amino acid hormone, peptide hormone, which is called as glucagon, and we all know that glucagon is hyperglycemic. Insulin is hypoglycemic, and you know cortisol also has the similar activity like that of glucagon. Means cortisol works generally antagonistic to insulin. NCERT I mentioned ചെയ്തിട്ടുണ്ട്. Cortisol, you know, cortisol is released from adrenal cortex, you know, zona glomerulosa, zona fasciculata, zona reticularis, zona glomerulosa releases mineralocorticoids, zona fasciculata glucocorticoids, and zona reticularis sex steroids, androgenic sex steroids generally. അപ്പോൾ ഇത് adrenal cortex എന്നാണ് വരിക, and it works antagonistic to insulin. Aldosterone is mineralocorticoid of adrenal gland, and it helps in mineral homeostasis, mineral balance and all, so that is not involved. But glucagon and cortisol, it works opposite to the manner insulin has effect on carbohydrate, protein and fat metabolism. So for question number 167, answer is 4, both 1 and 2. അപ്പോൾ 167, which of the following regulates carbohydrate, protein and fat metabolism in a manner opposite to that of insulin is both 1 and 2. You have to go through your NCERT carefully. Compare the characters. ഓരോ ലൈനും ഓരോ പോയിന്റാണ്. Okay, so for 167, 4 is the answer. With that, I go to 168. 168 says, at what point the secondary oocyte complete meiosis? We know spermatogonial cells, they form primary spermatocyte. Primary spermatocyte will undergo meiosis 1, resulting in the formation of one secondary oocyte. Spermatogonial cells, they are associated with spermatogenesis. Here the question is oocyte. So generally oogonial cells, they will form primary oocyte. Primary oocyte will undergo meiosis 1, resulting in the formation of one secondary oocyte and one polar body. And this secondary oocyte will be arrested there, and it will be entering to the meiosis 2 only after fertilization. This is what we know. So here, at what point the secondary oocyte complete meiosis? What? When the acrosomal enzyme break down the jelly coat? No. When the LH trigger ovulation? No. Ovulation is generally due to LH surge. Here the question is when it will complete, when secondary oocyte will complete oocyte. Secondary oocyte is going to complete the meiosis, means meiosis 2, to form ootid or ovum. So it's not during ovulation. Ovulation, secondary oocyte comes out. So after it is penetrated by the sperm cell, yeah, that is the answer. Answer is 3. After the fusion of sperm and egg nuclei, no, it's not like that. So answer is after it is penetrated by the sperm cell. So for 168, 3 is the answer. With that I come to 169. Each seminiferous tubule is lined by, seminiferous tubule of the testis. It has Sertoli cells, otherwise called sustentacular cells, otherwise they are called nurse cells. And they have spermatogonia. And the development stage and all, it occurs exterior. So each seminiferous tubule is lined by spermatogonia, yes. Primary spermatocyte, no. Sertoli cells, yes. So 169 answer is 4. This is direct, a simple question. This is Sertoli cells which will provide nourishment to the developing sperm cell. So it's a very direct question. Primary spermatocytes, secondary spermatocytes, spermatids, they are all at the different stages. Here it is lined inside by nanu choദിchirunne. So answer is spermatogonia and Sertoli cell. In that sense, both 1 and 3. So answer is 4. With that, let me go to question number 170. So if you look at question number 170, periodic abstinence works on the principle of. So when you say periodic abstinence, so no delivery of the sperm or the act of copulation during ovulation. That is the major concept here. So periodic abstinence works on the principle of preventing implantation, no. Preventing ovulation, no. Preventing fertilization, yes. Because if ovum is available, fertilization is bound to happen if there is delivery of sperm. Our sperm cell ഉണ്ടെങ്കിൽ അവിടെ ഫെർട്ടിലൈസേഷന് സാധ്യത കൂടുതലാണ്. അപ്പോൾ അത് പീരിയോഡിക് ആബ്സ്റ്റിനൻസ്. പ്രിവെന്റിങ് ovulation and ovulation is never fertile. അത് പർപ്പസേ അല്ല. നാച്ചുറലി ovulation ഒരു നോർമൽ ഹെൽത്തി ഫീമെയിലിൽ നാച്ചുറലി അത് പ്രിവെന്റ് ചെയ്യാൻ പറ്റില്ല. Artificially by taking medicines and all, that is secondary. But here the question is very simple. Periodic abstinence works on the principle of, so that no coitus, no fertilization during this stage when ovum is available. So answer is 3, 170. 3 is the answer. With that I come to 171. 171, in case of a couple where the male is having a very low sperm count. In case of a couple where the male is having a very low sperm count, which of the following technique will be suitable? So, gamete intrafallopian transfer, intracytoplasmic sperm injection, intrauterine devices. So it is artificial insemination, right? That is the best way, because the individual is producing low sperm, very low sperm. So sperm is available. അപ്പോൾ നോർമൽ കൗണ്ട് ഇല്ലയെന്നേ ഉള്ളൂ. അപ്പോൾ ജനറലി ഫോർ ഫെർട്ടിലൈസേഷൻ artificial insemination can be a general technique that is used for a couple where the male is having a very low sperm count. So for 171, 2 is the answer. With that I come to 172. Question number 172 says, find the mismatch. Agriculture started, agriculture started 10,000 years ago. ശരിക്കുള്ള ഇത് എവല്യൂഷൻ ചാപ്റ്റർ എന്നുള്ള ക്വസ്റ്റ്യൻ ആണ്. അഗ്രികൾച്ചർ സ്റ്റാർട്ടഡ് 10,000 years ago എന്നാണ്. ഇവിടെ എത്ര എഴുതിയേക്കുന്നത്? 1000 years ago എന്നാണ്. അപ്പോൾ അത് തന്നെയാണ് മിസ്മാച്ച്. Agriculture started 10,000 years ago. You can see your NCERT, human evolution, towards last paragraph. 18,000 years ago, you can trace cave painting. 10,000 years ago, you can see agriculture. Homo habilis brain size 650 to 800 ccf. Java, Java. Fossil discovered from Java. Java man. Neanderthal man, 1400 brain size. That is correct. Neanderthal man is the evolutionary stage of human where animal hide was used to cover the private body parts. Animal hide, you see that burying their dead ones, rituals. So Neanderthal, it is 1400 cc brain size. So when we compare all 172, find the mismatch. 1 is the answer. Agricultural started 10,000 years ago. Here it is 1,000 years ago. So 172, 1 is the answer. With that, let us go to 173. Select the false one regarding religious view on evolution. അപ്പോൾ ഏതാണ് തെറ്റ് ഫ്രം റിലീജിയസ് ലിറ്ററേച്ചർ ഓർ റിലീജിയസ് പോയിന്റ് ഓഫ് വ്യൂ എന്നാണ് ചോദിക്കുന്നത്. നമുക്ക് നോക്കാം. Religious literature tells us about special creation. Living things are created by divine power. That is correct. Father Soares was one of the greatest supporter of theory of special creation, that all living forms, whatever we see in the present world, ഈ ലോകത്തുള്ള എല്ലാ ലിവിങ് ഫോംസിനെയും ദൈവമാണ് ഉണ്ടാക്കിയിരിക്കുന്നത്. സോ religious literature tells about special creation, that living things are created by divine power, God. So that is correct according to theory of special creation. According to this, all creatures we see today were formed as such. Yes, very much. ഉണ്ടാക്കുമ്പോൾ തന്നെ ഇങ്ങനെയായിരുന്നു എല്ലാ ജീവജാലങ്ങളും, ഇതേപോലെയായിരുന്നു കാണാൻ ആദ്യ കാലഘട്ടം മുതൽ ഇന്നേ വരെ. അപ്പോൾ ആദ്യത്തെ ശരിയാണ്, രണ്ടാമത്തെ ശരിയാണ്, മൂന്നാമത്തെ ബയോളജിക്കൽ ഡൈവേഴ്സിറ്റി ഇൻക്രീസ് സിൻസ് ദ ക്രിയേഷൻ ഓഫ് ലൈഫ്. ഇറ്റ് ഈസ് നോട്ട് ലൈക്ക് ദാറ്റ്. റിലീജിയസ് ലിറ്ററേച്ചർ ടെൽസ് the form of life that you see now and the form of life that was there first ആദ്യ കാലഘട്ടത്തിൽ ഒക്കെ ഇവരുടെ ഒക്കെ സാന്നിധ്യം ഉണ്ടായിരുന്നു എന്നാണ് പറയുന്നത്. പക്ഷേ ഇവിടെ സ്റ്റേറ്റ്മെന്റ് ബയോളജിക്കൽ ഡൈവേഴ്സിറ്റി ഇൻക്രീസ് സിൻസ് ദ ക്രിയേഷൻ ഓഫ് ലൈഫ്, ദറ്റ് ഈസ് ഡാർവിനിസം, ബിക്കോസ് natural selection played role and survival of the fittest. So all these things, this is not as per biological. Earth is only 4,000 years old to an extent that also can be related to religious activities. So the best out of 4, 3 is not according to religious view on evolution. So for question number 173, 3 is the answer. With that, I go to 174. This is direct question. Malignant malaria is caused by Plasmodium falciparum. ഇത് ഡയറക്റ്റ് ആണ്. ബാക്കിയുള്ള ഡിഫറെൻറ്റ് സ്പീഷീസ് ഓഫ് malarial parasite, Plasmodium vivax, Plasmodium ovale, Plasmodium malaria and Plasmodium falciparum. And you know that malignant malaria is caused by Plasmodium falciparum. ആണ് അത് കോസ് ചെയ്യാം. വളരെ ഇമ്പോർട്ടൻ്റ് ആണ്. Malaria, Dr. Ronald Rose was awarded Nobel Prize. And it is a digenic parasite, one in female Anopheles mosquito and in human. And in human, the protozoan, Plasmodium, it thrives in hepatocytes and erythrocytes. So, human is a sexual phase, mosquito is sexual phase. So, this is a very popular question also, direct question also. Malignant malaria is caused by, answer is Plasmodium falciparum. With that, I come to question number 175. So, question number 145, this is from Human Health and Disease. Match the item in the column 1 with that of column 2. Opioid, opium, morphine. Right? Morphine. It's a pain reliever also. So A is 3. IgA provides natural passive immunity. IgA is also called secretory antibody because it is present in colostrum, mother's milk. So post-parturition, natural passive immunity is provided by IgA. അതായത് ജന്മം കിട്ടിയതിന് ശേഷം ആ കുട്ടിക്ക് പാല് അമ്മ ഊട്ടുമ്പോൾ ആ പാലിൽ IgA ആൻറ്റിബോഡീസ് ഉണ്ടാവും. ഇനീഷ്യലി കൊളസ്ട്രോം ആയിരിക്കും. കൊളസ്ട്രോം ഈസ് ഓൾസോ റിച്ച് ഇൻ IgA ആൻറ്റിബോഡി. Immunoglobulin alpha. So it's a globular protein. Apo you can find B4. Cocaine. Cocaine, we know it is erythroxylone coca. It is a stimulant. So C is 2. Stimulants. They stimulate the CNS, nervous system. And stimulants have a lot of, not only cocaine, there are other stimulants also, you know, and they have their own medical application. Alpha interferons, you know, they can be used in the treatment of cancer. So cancer treatment, so D is 1. So 3, 4, 3, so this cannot be the answer, this cannot be the answer. 3, 4, 3, 4. And C is cocaine, which is stimulant 2. So this is the answer. So answer for question number 175 is 2. Opioid, which is morphine, a painkiller, pain reliever. IgA, which is present in colostrum, that is providing passive immunity. Cocaine, you know, it's a stimulant. Alpha interferon for cancer treatment. The question is from Human Health and Disease. Direct question also. So 175 2 is the answer, and I come to page 176. 176, the basic unit of class. This is also a direct question and a very popular question. The basic unit of classification is species. You know, species are group of closely related individuals which can freely interbreed in nature and should produce fertile offspring. So there are so many species under the genus, and you know that in a so different population organisms. So the basic unit of classification is species. They form a group which are closely related. They freely interbreed in nature. Not only breeding is the criteria. അവരിൽ നിന്നും ജനിക്കുന്ന കുഞ്ഞ്, when that fellow become adult, when that fellow participate in reproduction, still in that case also the adult one will be fertile. So fertility is one of the criteria for the propagation of species. So basic unit of classification is species, where you can see group of closely related individuals which can not only freely breed in nature but must also produce fertile offspring. Very, very important. Fertile offsprings. And the basic unit of classification is definitely species. So for 176, 1 is the answer. With that I go to 177. Who discovered triple helical structure of collagen? We all know triple helical structure of collagen was discovered by G.N. Ramachandran, and the plot of the protein in which he has given the position for collagen, that is a Dr. Ramachandran plot. A good amount of work was also seen in India in Madras University campus. Dr. G.N. Ramachandran. Pauling for in the field of chemistry also, biology also. Berzelius also in the field, and Einstein, everybody knows. So here collagen triple helical structure was proposed by G.N. Ramachandran, and the plot is where the collagen structure is situated. It is called Dr. Ramachandran plot. So 177.1 is the answer. With that I go to 178. 178, identify the wrong statement regarding the protein structure. You know protein structure, primary structure, secondary structure, tertiary structure, and the most complex of all with quaternary structure, having more than one polypeptide and must have definite structure in all the proteins. Identify the wrong statement. Identify the wrong statement which is not correct. Identify the wrong statement regarding protein structure. Peptide bond is present in primary structure. Yes. Primary structure is linear structure, simple structure, generally linear structure, simple structure, where two amino acids are bonded by peptide bond. And when you go to secondary structure, you can also see hydrogen bonds. Gradually, when you see hydrogen bond is present in secondary structure, yes, that's correct. Tertiary structure give catalytic activity to protein. There are so many examples. Alpha helix and beta plated are primary structures. No, they are secondary structures. So this is wrong. So alpha helix and beta plated are secondary structures. Secondary proteins are further classified into alpha helical and beta plated. So for question number 178, 4 is the answer. With that, I come to question number 179. Question number 179, dihybrid phenotypic ratio is. So here different kinds of ratio are given. Basically we know, if you take up Mendelian, if you see monohybrid, Mendelian monohybrid phenotypic ratio, Mendelian monohybrid phenotypic ratio, if you see phenotypic ratio, it is 3:1. And we know that Mendelian monohybrid genotypic ratio is 1:2:1. Dihybrid phenotypic ratio is 9:3:3:1. And dihybrid genotypic ratio is 1:2:2:4:1:2:1:2:1. So here you know dihybrid phenotypic ratio is 9:3:3:1. Idum direct question aana. Dihybrid phenotypic ratio is, right? Dihybrid phenotypic ratio is 9:3:3:1. Okay. With that, I come to question number 180. 180, if you see ZWZZ. So you know that if you go with the classification can be XX, which is a female, XY, which is male. Here male is heterochromatic. Classical example you can say human, Drosophila. So they all come under this category. Then you would have studied about XX, XO, insects. For example, grasshopper. So here this is female, which is homogametic, and this is male, which is heterogametic. Then it is ZWZZ. Here the female is heterogametic and male is homogametic. So here you can see, say humans, classical example, humans, Drosophila, they all come under this category. Female is homogametic, male is heterogametic. Certain insects, you can take up grasshopper, classical example. ZW, female is heterogametic, male is homogametic. You can see this in birds and reptiles. Birds and reptiles come under this category. And if you see ZOZZ, where ZO, here it is female who is heterogametic, and this is male which is homogametic. Classical example, you get certain moths and butterflies, certain moths, etc. They come under this category. So here female is heterogametic. Now the question here, in ZWZZ type of sex determination, here this is female and this is male. very clearly we should understand. This is female, this is male. Females are heterogametic, that is the answer. Males are homogametic, heterogametic. Females are homogametic, not possible. Females and males are heterogametic, not possible. Here female is heterogametic, and this kind of things you can see in birds and reptiles. So classical example 181 is the answer. So for question 181 is the answer. With that I come to 181. If the blood group of one parent is O group, so here it is very clearly asked that if the blood group of one parent is O, so one parent is O, and their offspring is of B group, so offspring B group, kuttida blood group B group. What are the possible blood group of their parent? Okay, നമുക്കൊന്ന് നോക്കാം. ശരി, അങ്ങനെയാണെങ്കിൽ ക്രോസ് വെച്ച് നോക്കുകയാണ് വെച്ചാൽ, അപ്പോൾ ഒരു പേരന്റ് തന്നിട്ടുണ്ട്. സപ്പോസ് ദ അതർ പേരൻസ് ബ്ലഡ് ഗ്രൂപ്പ് ഈസ് എ. നമുക്ക് ഹെട്രോസൈഗസ് എടുക്കാം, അപ്പോൾ പോസിബിലിറ്റി കൂടുമല്ലോ. അപ്പോൾ ഇപ്പോൾ O into A is always A. അപ്പോൾ ഇവിടെ നോക്കുമ്പോൾ ദിസ് ഈസ് യുവർ P1. ഫ്രം ഹിയർ G1 will come, which is I, and here there will be two types of gametes, which is IA and I. And in F1, if you see F1, so this is I, this is IA, so this will be IA, I, and this will be O blood group. അപ്പോൾ ഇത് O blood group, അപ്പോൾ ഇത് A ആയിരിക്കും, അല്ലെങ്കിൽ O. അപ്പോൾ അടുത്ത പേരൻറ്റിൻ്റെ ബ്ലഡ് ഗ്രൂപ്പ് A ആണെങ്കിൽ ഒരിക്കലും B ബ്ലഡ് ഗ്രൂപ്പുള്ള ചൈൽഡ് വരാനുള്ള സാധ്യത ഇല്ല. അപ്പോൾ ആദ്യത്തെ ഓപ്ഷൻ അല്ല. ഓക്കെ, രണ്ടാമത്തെ ഓപ്ഷൻ, സപ്പോസ് ഹിയർ ഇഫ് ദ ബ്ലഡ് ഗ്രൂപ്പ് ഓഫ് വൺ പേരന്റ് ഈസ് O and the offspring is B, what are the possible blood groups of other parents? Right? So here if you see, next case, so one is an other, it is IBI, or this is the case. അപ്പോൾ ഇത് ഇതിൻ്റെ ഒപ്പം ചെയ്ത് കഴിഞ്ഞാൽ definitely B will come, but the child can be O. Right? അപ്പോൾ അതുമല്ല A, B or O, Aയിൽ വരില്ല എന്നാ ഒരു കാര്യം ചെയ്യും. Suppose one parent is O, other parent is B. Assume. So in this case also, one parent നമുക്ക് കിട്ടിയിട്ടുണ്ട്. അപ്പോൾ ഇത് ഇങ്ങനെ വരും, ഇത് ആസ് ഐ ടോൾഡ് യു ഇങ്ങനെ വന്ന കോമ്പിനേഷനിൽ ഇത് I, ഇത് IB, ഇത് I. So there is a chance that the child can be. അപ്പോൾ ഈ കോമ്പിനേഷൻ ഒന്ന് കറക്റ്റാണ്. ഇനി ഇതാണെങ്കിലോ, സപ്പോസ് ദ അതർ പേരന്റ് ഈസ് AB, so this will be IA, IB. അങ്ങനെയാണെങ്കിൽ ഇത് ഇങ്ങനെ, this is your P1, G1, ഇത് IA, IB. In this case also, when I do the cross, either I, either IA, either IB. Suppose paternal matter. So this is the best possible combination. The other parent can be either B or AB. In both the cases, the child can be B blood group. ഈ ഒരു കോമ്പിനേഷനിൽ ഇത് ആണെങ്കിൽ B ആവാം, പക്ഷെ അടുത്ത പേരന്റ് O ആണെങ്കിൽ ഒരിക്കലും B വരില്ല, കാരണം ചൈൽഡ് O ആയിരിക്കും. So the best possible answer is 4. കാരണം ഇവിടെ ചോദ്യം എങ്ങനെ, if the blood group of one parent is O group and their offspring is of B group, what are the possible blood groups of other parent? കോമ്പിനേഷൻ കൊടുത്തിരിക്കുക. A or O പറ്റുമോ? ഇല്ല. B or O B ആണെങ്കിൽ ശരി, പക്ഷെ O ആണെങ്കിൽ പറ്റുമോ? ഇല്ല. A ആണെങ്കിൽ പറ്റില്ല, B or അങ്ങനെയല്ല. B or AB ആണ് അടുത്ത പേരന്റ് ആണെങ്കിൽ ഉറപ്പായിട്ടും ഓഫ്സ്പ്രിങ്ങിന് B blood group വരാനുള്ള സാധ്യത ഉണ്ട്. So for question number 181, 4. is the answer. With that, I go to question number 182. During DNA replication, deoxyribonucleoside triphosphates are dual purpose. So, for deoxyribonucleoside triphosphate, which can also be written as DNTPs, deoxyribonucleoside triphosphate of DNA include DATP, DGTP, DCTP and DTTP. These are the deoxyribonucleoside triphosphates. So this dual purpose, what is a dual purpose? Let me tell you. Consider this is DATP. DATP will be converted to DAMP, deoxyriboadenosine monophosphate. Using this, you can make DNA. So this is the substrate to make DNA. And idunnu baaki rendu inorganic phosphate ille, 2 inorganic phosphate, and this 2 inorganic phosphate will provide energy. Appo dual function undu deoxyribonucleoside triphosphate. One, it acts as substrate. Rendu, it provides energy. So it acts as substrate and provide energy. That is the answer. 182. Act as catalyst. Catalyst allowed. And substrate, no. Act as substrate and ribozyme. What are ribozymes? Let me tell you. So if you ribozymes are RNA with enzymatic or catalytic activity. So catalytic or enzymatic activity. RNA with enzymatic or catalytic activity is ribozyme, or you can also say that these are non-proteinaceous enzymes, proteins allatha enzymes. Ribonucleotides include ATP, GTP, CTP and UTP. So this is our answer. So for question number 182, during DNA replication, deoxyribonucleoside triphosphates are dual purpose. What is that? Act as substrate and provide energy. So for 182, 1 is the answer. With that, I come to 183. 183 says, what property of genetic code allows the use of several codons for a particular amino acid? This is also a direct question. And we all know that that property where several codon for a particular amino acid is called degeneracy. One amino acid onil koodal codon. For example, if you take up the example, for example, you see codons of say valine. That will be GUU, GUC, GUA, GUG. Serine, if you take up serine, UCU, UCC, UCA, UCG, AGU, AGC. So you see, there are only 2 amino acids, methionine and tryptophan. Only 2 amino acids, methionine and tryptophan, do not have degeneracy. 2 amino acids, methionine and tryptophan, degeneracy illa. Methionine and tryptophan, they have only one codon. Codon of methionine is AUG and codon of tryptophan is UGG. These are the only 2 amino acids which are codon specific. Generally what happens, codon is amino acid specific. Amino acid is not specific to codon except for methionine and tryptophan. Methioninum, tryptophanum ഒഴികെ ബാക്കി എല്ലാ കേസിലും codon is amino acid specific. Amino acid is not specific to codon. So methionine and tryptophan, they don't show degeneracy. Universality, yes. Genetic code is almost universal. Genetic code is almost universal. Non-overlapping, non-ambiguous. Yes, they are unambiguous. Means a particular codon always code for particular specific amino acid. Or a particular codon or a particular amino acid nu vendi maatrame code cheyu. ഒരു പർട്ടിക്കുലർ കോഡോൺ ഒരു പർട്ടിക്കുലർ അമിനോ ആസിഡിന് വേണ്ടി മാത്രമേ കോഡ് ചെയ്യൂ. സോ അൺആംബിഗ്യസ് അല്ലെങ്കിൽ നോൺ ആംബിഗ്യസ്. പക്ഷെ ഇവിടെ ചോദിച്ചേക്കുന്നത് വാട്ട് പ്രോപ്പർട്ടി ഓഫ് ദ ജനറ്റിക് കോഡ് അലൗസ് ദ യൂസ് ഓഫ് സെവറൽ കോഡോൺസ് ഫോർ എ പർട്ടിക്കുലർ അമിനോ ആസിഡ്? ആൻസർ ഈസ് degeneracy. So for question number 183, 2 is the answer. With that I come to question number 184. Production of large holes in Swiss cheese is by, we know it is by Propionibacterium shermani. So answer is 1. It's not Saccharomyces cerevisiae or Penicillium roqueforti or Aspergillus niger. And you know this, that these large holes over the Swiss cheese is because of production of large amount of carbon dioxide. So answer, production of large holes in Swiss cheese is by Propionibacterium shermani. This is a direct question. There is a direct question. So 184. 1 is the answer. With that I come to question number 185. Protected areas are example. Protected areas are those areas where you conserve the species within their habitat without creating any artificial habitat or restricted habitat that is created by human, such as zoos and all. No, you don't do that. So it is in situ conservation. So you know that protected areas are examples of in situ conservation. Protected areas are examples of in situ conservation. Ex situ, you know, from their original habitat to man-made habitats where we conserve them, we preserve them. Cryopreservation is using liquid nitrogen, it's minus 196 degrees Celsius. Greenhouses, no, it is not like that. So for question number 185, where it says protected areas are examples of, answer is in situ conservation. So 185, 1 is the answer. With that I come to question number 186, which is section B. Answer any 10 questions. Section B. Answer any 10 questions. Let us see. Which of the following is not seen in saccoglossus? You know, saccoglossus and belanoglossus are the examples of phylum Hemichordata. Hemichordata was previously kept under Chordata, but now it is an independent phylum. Phylum Hemichordata, whose body is divided to proboscis, head and trunk. So the body has tripartite body organization, correct? Proboscis, collar, trunk. Stomachode is there. Yes, it is not a notochord. A rudimentary notochord? No. Proboscis gland for X? Yes, that is present. So answer, it's a very direct question. 186. So 186 here, 3 is the answer. That which of the following is not seen in saccoglossus? Answer is a rudimentary notochord. It has stomochord, not notochord. So 186. 3 is the answer for phylum Hemichordata. Example here given is saccoglossus. Tripartite body organization, proboscis, collar, trunk. That's how it is. So what is, okay, so with that I come to question number 187. 187, what it says? What is common among amylase? You know amylase, classical example salivary amylase, which is otherwise called tyalin, or it can be pancreatic amylase. Amylase, these are carbohydrate digesting enzymes. That's what I want to tell you. Amylase, these are carbohydrate digesting enzymes. Renin, RENNIN, so that is protein digesting enzyme, mainly active in infants. And trypsin, you know, trypsin is also called universal enzyme. So trypsin is protein digesting enzyme. Amylase is carbohydrate digesting enzyme. Renin is protein digesting enzyme. Renin act on casein to convert paracasein, and paracasein is then converted to calcium paracaseinate by renin, but calcium paracaseinate is converted to protease, peptones and polypeptides by pepsin. Okay. So what is common among them? They are all proteins? Yes, that is the answer. They are all enzymes. And most of the enzymes, they are proteins. Most of the enzymes, these are proteins. They are all proteolytic enzymes? No. Amylase is carbohydrate digesting enzyme. Amylase is carbohydrate digesting enzyme. Renin is proteolytic. Trypsin is also proteolytic. So this is not the option. All are produced in stomach? No. Salivary amylase is produced in mouth. Pancreatic amylase from pancreas to pancreatic juice and then to duodenum. No, it is not correct. All need a pH less than 7? No. No. Renin and renin, it works at pH 1.8. Amylase 6.8. Right? Trypsin 7.8. So no, it is not correct. So for question number 187, 1 is the answer. With that, I go to question number 188. The center which can moderate function of respiratory rhythm means pons. In textbook, it's very clearly written. Pneumotoxic centers are present in pons. So this is a direct question. The center which can moderate the functions of respiratory rhythm center is present in pons varoli region or pons region of brain. Okay. Thalamus has upper part which is epithalamus with pineal gland, hypothalamus with pituitary, and thalamus includes your diocele and it is lined by ependymal cells. You know that. So 188, 2 is the answer. With that, I come to 189. 189. What is incorrect about Periplaneta americana means cockroach? Excretion by malpighian tubules, that is correct. If you go to structural organization of animals, you can read through in your NCERT also. It is very clearly written that Periplaneta americana excretion is by malpighian tubule. Hemolymph delivers oxygen to the tissues. That is wrong. Because you know the respiratory system in cockroach, it is made up of trachea, and the opening exterior, it is called spiracles, and the oxygen air enters through the spiracles to the tracheal tubes, reaches the cell. So that is incorrect. Hemolymph generally transport of nutrients, removal of waste, these kind of things major. Incomplete metamorphosis, mosaic vision, you know that. Insects. So for 189. 2 is the answer, and I come to question number 190. Question number 190, what it says? During muscle contraction, the length of actin, we know, will never get reduced. So A band, it is a direct question. During muscle contraction, the length of all the following gets reduced except A band, actin band. So you know that it is central band which does not get reduced. The other filaments, they come nearby and move away. So it is direct question. 190. 190 answer is 2. With that, I come to question number 191. So question number 191, Reisner membrane separates. This is a direct question. You know that Reisner membrane separates scala vestibuli and scala media. Tympanic cavity and pharynx, no. Round window and oval window, no. Scala media and scala tympani, these are separated by basilar membrane. Basilar membrane. They are separated by basilar membrane. They are separated by which membrane? Basilar membrane. So, Reisner's membrane. So here the question is Reisner's membrane. Reisner's membrane separate scala vestibuli and scala media. So 191, 1 is the answer. With that, I go to 192. 192. Which of the following are not steroid hormones and yet do not act through a second messenger system? Let us see. Estrogen and progesterone, these are steroid hormones. Insulin and glucagon, protein or peptide hormones. So these are your generally protein hormone. Calcitonin and PTH, these are also generally protein hormone. T3 and T4, these are amine hormones. Okay, so answer is your 4. Which of the following are not steroid hormone? This is not steroid hormone, yet do not act through a second messenger system. Answer is T3 and T4. So that is how we understand 192. You have to follow your NCERT thoroughly. With that, I come to 193. Among the following, the number of edible freshwater fish. നമുക്ക് നോക്കാം. Pomfret is marine water. Pomfret is a marine water edible freshwater fish. The question is edible freshwater fish. Pomfret is marine water. Katla. Rohu is labio rohita. This is freshwater. This is freshwater. So pomfret is marine water. Katla is freshwater. Rohu is freshwater. Sardine is marine water. Sardine is marine water. Hilsa Elisha, a Bay of Bengal region. This is very popular fish in Bengal. So this is also a marine fish. Common carp, it is freshwater fish, edible. So among the following, the number of edible freshwater fish, 1, 2, 3, answer is 3. So for question number 193, option 2 is the answer. That means 3 of them are freshwater edible fish. With that, I come to question number 194. Question number 194 says, which of the following is at its peak level during luteal phase of menstrual cycle? You know that hypothalamus releases it in a very simple way, if you can. So your hypothalamus, in a very simple way, you can see the hypothalamus, it releases. Hypothalamus releases your GnRH, and this acts on adenohypophysis or anterior pituitary. Anterior pituitary will release LH, luteinizing hormone, which will generally act on corpus luteum. Corpus luteum of ovary, and corpus luteum will release what is called as progesterone. So during LH phase, you can see plenty of progesterone. During LH phase, you can see plenty of progesterone. So which of the following is at its peak level during the luteal phase of menstrual cycle? Answer is progesterone. Estrogen is under follicular phase. FSH will act on Graafian follicle, which will release estrogen. LH and FSH, these are hypothalamic hormone. So for 194, 2 is the answer. With that, I come to question number 195. 195, industrial melanism is an example of. So first option, disruptive selection. So you know, if you go through Hardy-Weinberg equilibrium, the last of the factors which affect is natural selection, of which the third one with 2 peak, it is disruptive selection. Classical example, sickle cell anemia RBC and biconcave RBC people surviving simultaneously. So industrial melanism is not an example of disruptive selection. Inheritance of acquired characters are not inherited. Directional selection, yes. That means, that is the answer. You can see the peak, it shift from one population to other population. Initially, it was white population which was dominating and dark population was least. After Industrial Revolution, the dark population thrived, white reduced. So it is directional natural selection. So for question number 195, 3 is the answer. With that, I come to question number 196. Question number 196 says, in which of the following groups all of them are polysaccharide? Let us try to understand. So this is a direct question. Glycogen is a polysaccharide. Sucrose is a disaccharide, maltose is a disaccharide. Okay, sucrose is made up of glucose and fructose. And this is alpha 1,2 glycosidic bond. Maltose, it is glucose plus glucose, and this is alpha 1,4 glycosidic bond. Glycogen is a polysaccharide, glucose is a monosaccharide, sucrose I have already mentioned, it's a disaccharide. Maltose is a disaccharide, glucose. Lactose is disaccharide, galactose plus glucose, beta 1,4 glycosidic bond. This is also a disaccharide. Glycogen is a polysaccharide, cellulose is a polysaccharide, starch is a polysaccharide. Answer is 4. So for 196, 4 is the answer. With that I come to question number 197. Female heterogamety, which is say for example ZW female and ZZ male. Female heterogamety is common in the following organisms: human and Drosophila. Male heterogamety can be seen because they have XX, XY system. Grasshopper and cockroaches, it is XX, XO. Birds and reptile is the answer, because this type where female heterogamety, you can see male homogamety, female heterogamety and male homogamety can be seen in birds and reptiles. Okay, with that I come to question number, so for 197, 3 is the answer. 198, identify the set of codons which do not have anticodons. We should know stop codons do not have anticodons. This is very, very important. Rest all have anticodons. So UAA, you know, which is otherwise called ochre, UAG, which is otherwise called amber, UGA, which is otherwise called opal. These are the stop codons which do not have anticodons. Stop codons do not have tRNA, and stop codons do not code for any amino acid. So here just identify the stop codons. You see 3 is the answer: UAG, UGA, UA. So for question number 198, 3 is the answer. Question number 199, match the following. Let us see. Citric acid, Aspergillus niger. So direct question. A is 3. Pectinase, bottle fruit juice. So B2. Against life, antibiotics. So C is 4. Staphylococcus aureus, Fleming's work for penicillin. D1. So 3241, 3241, answer is 2. So for question number 199, 2 is the answer. And I come to the last question. Select the incorrect match. Aravali Hills, this is incorrect because Aravali Hills you can find majorly in Rajasthan, Haryana, certain part of Delhi, ending in Ahmedabad. Khasi and Jayanti Hill, Meghalaya. Sarguja, Madhya Pradesh, yes. When it was Sarguja district, it was there in Madhya Pradesh. Now it is called Chhattisgarh. But basically it was registered first in Madhya Pradesh. Western Ghat, Karnataka, Maharashtra, correct? Western Ghat you can find in Maharashtra, Karnataka, Goa, Kerala. So best out of 4 when you take, answer is 1. So for 200, answer is 1. So that concludes our question paper. I hope you would have scored maximum marks for next exam. Keep learning. Follow your NCERT. God bless you. Thank you.